Okay, this is chapter 14 of the immune system. This is going to be an overview of the entire immune system and then some specifics about our non-specific defenses, our first and our second lines of defense. Our defense system can be broken down into two different systems, our innate or nonspecific and our acquired specific immunity. We have three lines of defense. These are your first, your second, and your third lines of defense. These three lines of defense can be divided into our innate and our acquired immunity. Your defense system is a very complex system. It's not necessarily complicated, but there are a lot of things going on that allow these systems to take on some very specific jobs to keep the body from becoming invaded by microbes that could otherwise cause damage. This graph here basically is an overview of the entire host defenses. You'll see that we've broken it down into our innate and our nonspecific immunity, and that innate immunity is broken down into our first and second lines of defense. Your first lines of defense are going to be your physical and chemical and genetic barriers. Your second line of defense are going to be some mechanisms and some other systems that are going to be activated that are going to allow for the further protection by our innate immunity. This is also a non-specific defense. The second line of defense is going to include inflammatory responses, the release of interferons, phagocytosis, and a cascade of events called complement. Our acquired, or our third line of defense is going to be our acquired specific immunity. This can be divided into our naturally acquired and our artificially acquired immunities, and these are going to give rise to our antibodies, our B cells, and our T cells, as well as some accessory organs. Most of our third line of defense is going to be covered in chapter 15. Chapter 14 is going to deal mostly with our first and our second lines of defense. And we'll get into the third line of defense in chapter 15. Your innate immunity is a nonspecific response to any kind of invader or tissue injury. This is going to include your first and your second lines of defense. Your acquired immunity is going to be specific. It's directed towards a particular microbe, and it is only going to be activated once you have been infected with a particular organism. This is why we call it an acquired immunity. You have to be sick at least one time before you have a very significant third line response. In our innate immunity is always on guard. It has no time off, if you will. The physical and chemical barriers, the genetic components of our innate immunity are going to be those things that are going to constantly be working. Inflammation, phagocytosis, these do not improve with repeated exposure. When we get to our third line of defense, you're going to see that the more we are exposed to a pathogen, the more sensitive our body becomes to it, and it can be more aggressive and at killing it. So it does improve with repeated exposure. Your innate immunity is what it is, and it will never get any better. Your innate immunity is going to be involved in inflammation and phagocytosis. These are major parts, major mechanisms of the second line of defense. Our first line of defense are going to be those barriers, things that are going to try and keep microbes out of tissues that are deeper tissues where you can end up having some significant tissue damage. These are going to include the anatomical or physical barriers, chemical barriers, as well as genetic components that are going to give us a particular genetic predisposition to an immune response. Our physical barriers are going to be things like your skin. Your skin is a very tough organ that is going to keep other microbes out. If you've got good, nice, healthy, intact skin, that is your best defense against the infiltration of microorganisms. Then you have cilia. The respiratory tract is lined with cilia. This is going to allow for the removal of organisms that work their way into the respiratory tract, and you can push those out. Mucus, all of our openings to the outside are going to be covered with a mucus membrane, and the mucus within that those membranes is going to have a lot of antimicrobial properties. So that mucus is going to be very effective at killing bacteria so it doesn't get any deeper into the tissues. Then you're also going to have body secretions like your urine, defecation, sweat. Those are all going to be things that allow for the release of microorganisms so that they don't get into the body. Your chemical barriers, these are going to include things like your sebaceous secretions. Your sweat is a chemical barrier. It carries a number of components in it. It has this very distinctive pH that can cause these bacteria to die before they ever get into a position where they can make you sick. We have an enzyme within our tears in our secretions called lysozyme, and this is going to be an enzyme that essentially breaks down the gram-positive cell wall, and so it's very effective at those upper respiratory infections, those infections that could be of the eye or the skin. You also produce lactic acid and have some specific electrolyte concentrations in the sweat that are going to make the bacteria unable to survive in those conditions. The skin has an acidic pH. It has fatty acid content to it that's going to increase, or excuse me, is going to decrease the pH, and the skin is relatively acidic and bacteria can't live in those low pH environments. The pH of the stomach. The stomach is lined with mucus that protects the stomach itself, but the stomach juices have a lot of hydrochloric acid in it, and those bacteria are generally not going to survive. Now, we do have bacteria that have been able to survive the stomach and make their way to the gastrointestinal tract. Once they get there, if they are not suited to live there, then the digestive juices as well as the secretion of bile into the intestines is going to take care of those organisms. Semen itself is antimicrobial. It's going to keep any organisms that may penetrate through the reproductive tract. The semen itself has some antimicrobial chemicals that can kill those. And then you have the pH of the vaginal tract is very low, sometimes as low as 3, and it can cause microbes to not be able to survive. We do have some genetic differences. Some of these, you have the hosts that are going to be genetically immune to specific diseases of other hosts. Some people have some sort of predestination for a predisposal, to immunity to specific diseases. There are populations of people that have a genetic predisposition for the immunity to HIV. They've been exposed a number of times and have yet begun to produce the symptoms of HIV. So they have some sort of a genetic immunity. This is going to be particularly true when we're talking about specific viruses. A good example of this, the HIV, but then you have a number of people, we've all been exposed to herpes simplex 1, the one that causes fever blisters, but many people don't get fever blisters because they have an immunity, and that keeps that virus under control. This does have some historical importance in that this genetic immunity has allowed for certain people to survive in certain geographical areas, that has allowed some populations then to increase and other populations to decrease. So it has some selective ability to allow for for populations to thrive in their environments that they live in. Your second and your third lines of defense are going to be those defense mechanisms that are going to take on any bacteria that have gotten into deeper tissues. They've penetrated the first line of defense, and now they're going to be into the deeper tissues. Your second line of defense is innate, meaning that you always have it, you're born with it. It's something that will be a defense against non-specific microbes. Any microbe that comes its way, it's going to try and deal with it. And there are 4 major phases in the second line of defense: inflammation, production and distribution of interferon, phagocytosis, and then complement. We're going to talk about each one of these individually, so don't panic that this is all you have to know about them. We're gonna talk about each one individually. Then your third line of defense, this is going to be an acquired immunity. This is something that we have to build up over time, and it is specific to a particular organism. This, your third line of defense, this is where you're going to begin to produce your B cells and your T cells. Those B cells and T cells are going to become activated, and those activated B cells then are going to produce antibodies. And you're going to have destruction then from that point on. Immunology is essentially the study of the way that the body responds to some sort of pathogen. It's going to be the study that looks at the body's second and third lines of defense most specifically. Any healthy immune system is going to be responsible for the surveillance of body tissues. It's going to be moving around. It's got cells within the immune response that are going to be surveying tissues to make sure that tissues don't become contaminated, and if they do, they're going to set forth some mechanisms that are going to destroy those. It's going to recognize anything that is foreign, that should not be in the body, that it says, wait a minute, this is not cellular tissue, I need to get rid of it. It also is going to be responsible for the destruction of any cell that it deems to be foreign. Now, when we talk about foreign matter, these are things that are not part of your host cellular structure. There are times, though, when the immune system actually kind of breaks down a bit and does not recognize your own tissues as the self tissues that it shouldn't mess with. And that's what we call an autoimmune disease. Unfortunately, we're not going to be talking much about autoimmunity. If you'll go to chapter 16, there's some extensive information on autoimmunity that you can read through. But anyway, anytime you have anything that the immune system does not recognize, it's going to think that it's foreign and it's going to destroy it. Now, this idea of self versus non-self. White blood cells that are the major workers, if you will, within the second and third lines of defense, they have to be able to distinguish white blood or self from non-self cells. Self tissues are going to be any tissue, any body cell of the host. It's going to be recognized by the immune system and say, this is normal, and I'm going to leave it alone. Then there is non-self tissue, and this is going to be any foreign material that is either microbial or it's going to be non-living in some form, and the body is then going to attack it. It's going to look at self versus non-self. If it recognizes it as self tissue, then there's going to be no reaction. If it doesn't recognize it as self tissue and it says that it's foreign, then it's going to employ some sort of mechanism like phagocytosis or inflammation to destroy those organisms, those foreign cells, and keep the body healthy. The white blood cells have the ability to distinguish self from non-self by looking at things that are called cell surface markers. These are going to be proteins or sugar molecules that are on the surface of the cell that will bind to receptors on some of these white blood cells, and they will say, hey, this is a cell surface marker, I recognize this, this is self tissue, and it's going to leave it alone. Your book talks about it as kind of the equivalent of facial characteristics. These facial characteristics are how I would recognize you. If somebody hides your face and I only see your body, I might have a difficult time recognizing who you are. But if they cover your body and just show your face, I'm going to know exactly who you are. So this is kind of the way that the white blood cells recognize self from non-self, is by these surface markers. This is going to allow the immune system to function correctly and not destroy self tissues and only destroy non-self or foreign tissues. There are a number of systems involved in the immune response, 4 of them in particular. You've got the reticuloendothelial system. You've got the extracellular fluid that's going to kind of work with between the lymphatic system and the reticuloendothelial system. You have the bloodstream, and you have the lymphatic system. These are going to be communication compartments that are going to be used within the cell to talk to other cells so that they can see where things need to be ramped up as far as the immune system is concerned. They can determine what kind of infection there is, and you can have a communication between these different body compartments. If you look here at this picture, you've got this this picture here that's got all of these labels here. Remember, in A&P you had to recognize these things called reticular fibers. This is part of the reticuloendothelium. This is going to be those reticular fibers that are in the endothelium of the cell itself, in the extracellular fluid. You have macrophages. These are going to be phagocytic cells. We're going to talk about these extensively here in a little bit. Then you've got the extracellular fluids. This is where you're going to have fluids leaving the lymphatic, going into the lymphatic capillaries, going into the and out of the blood capillaries. So this is going to be recycled through the lymphatic and the circulatory system. You have these cells called dendritic cells. These are very specialized cells. We'll talk about these again when we talk about the other cells. Then you've got your blood cells here, your capillaries that are going to be in these tissues in the body system. And then you have obviously your tissue cells. The reticuloendothelial system, or the RES, is essentially a network of connective tissues. It's going to provide a passageway that's going to be between tissues and organs that's going to allow for the movement of white blood cells within those structures, within that pathway. This is going to essentially be a filter. It's like a big filter. As fluids pass through this system, these reticular fibers are going to be embedded with macrophages, neutrophils, dendritic cells, all of these cells that are good at phagocytosis, and they're going to basically keep the fluids that run through that reticuloendothelial system from becoming infected. This system is going to essentially attack things that are going to attack the skin, the lungs, the digestive system, particularly the liver and the kidneys, lymph nodes, as well as the spleen and bone marrow. You'll notice here on the picture where it's got kind of purple tinting. That's where you're going to have a lot of this reticular tissue that's going to act as that filter. The organs of the reticuloendothelial system are going to be the thymus. The thymus is probably the most important immune organ that we have as a small child. This is where the white blood cells are going to mature. You also have lymph nodes. We'll talk about lymph nodes specifically when we talk about the lymphatic system. And then the spleen. The spleen is like a great big lymph node, but instead of using filtering lymph, it's going to filter blood. And so we've got that kind of system there. And then you've got your tonsils, your laryngeal and your pharyngeal tonsils. These are going to be filters for the upper respiratory tract. Again, this system contains a large number of white blood cells that are going to be phagocytic cells that are very good at phagocytosis and will kind of keep those body systems from becoming infected. The lymphatic system is probably the second of the compartments that does a lot the same thing as the reticuloendothelial system. It is a compartmentalized network. It does have vessels and cells as well as some accessory organs. This is going to reach most of the body parts. The RES, you'll notice that it was kind of isolated to different parts of the body. The lymphatic system, it goes everywhere, nearly everywhere. Its major function is going to provide some sort of return for extracellular fluid into the circulatory system. As that extracellular fluid goes into the lymphatic system, it's going to be taken into the circulatory system, recirculated, and then turned back into extracellular fluid. It also acts as a drain-off system for edema that happens during the inflammatory response. One of the characteristics of inflammation is swelling and edema, and that fluid has to go somewhere. So it gets drained off into the lymphatic system so it can return to the circulatory system and be excreted in whatever manner that it needs to be excreted. It's also going to give surveillance as well as recognition of foreign materials. It's also going to initiate the protection against these foreign materials. It's going to recognize these foreign bodies through cells that are called lymphocytes. We're going to talk specifically about lymphocytes when we talk about the different types of cells that we have. It's also going to activate phagocytes as well as antibodies that are going to recognize and destroy foreign microbes. Here's a good picture in your book. you'll see that the lymphatic system reaches nearly all of the parts of the body, even clear into the extremities. Here, I don't really care for this picture because it doesn't show any of the vasculature that goes up through the head and into the arms. But what it is showing you is that blood flows out of the heart through the arterioles, then through the venous circulation, back up into the heart. One of the things that you'll recognize is that this circulation goes in two directions. Lymphatic fluids go one direction. They go from the extremity all the way up to some sort of junction in the heart, in the veins near the heart. And so lymph then goes straight back into cardiovascular circulation, will be refiltered and be taken back out to the lymphatic circulation. If you look over here, this picture, you've got a large vein here, the subclavian vein, and you'll notice that it's got a number of lymph node clusters around it. These are called the axillary lymph nodes, and then you've got a lot of lymphatic material here of the mammary glands. These, you're going to see that we have a lot of lymphatic tissues, a lot of lymphatic ducts and pathways in areas like the breast, the hands, the feet, where we have a number of potential sites of contamination. The lymph is the fluid that goes through the lymphatic system. This is going to be a plasma-like liquid. It's going to be formed when you have certain blood components that move out of the blood vessels themselves into the extracellular spaces. Once they're in those extracellular spaces, they're going to diffuse then into lymphatic capillaries. Once in that lymphatic capillaries, now it becomes lymph. This is made up of mostly water. It is going to have a number of dissolved salts, a small amount of protein like antibodies and albumin, and then a lot of white blood cells. This is going to parallel plasma to a great extent without the red blood cells in it. You have the lymphatic vessels, which are going to be like your arteries in your veins. They are similar to some of the thin-walled veins that we have in the body. There are a higher number of these in the hands and the feet, and if you go back and look at that picture, you'll see that there are a lot of lymphatic vessels that go through the hands, the feet, and then around the areolae of the breast. These are because these are areas of frequent contact. We go barefoot, we touch things with our hands, babies suckle at the breast, and so you have a lot of environmental contact in those areas. Flow of lymph is going to be in one direction. It goes from the extremities to the heart, where it's going to dump into some sort of connection between the lymph vessel and the circulatory veins, generally at some point near the heart. You're going to have lymph ducts then are going to empty that fluid into the veins, where it will circulate into the heart, and as it goes through the cardiovascular circulation, it will be removed then and go back into the lymphatic system. Lymph is going to be moved through the body to the veins, much like venous blood flow. It's going to be via the contraction of skeletal muscles. Once those skeletal muscles contract, it's going to push that lymph up. Ducts are going to close, and it'll finally make its way up to the heart, where it will be dumped into the venous blood flow and be recirculated. Lymph nodes are very small, encapsulated structures. They remind me, all the pictures that I've seen remind me a little bit of edamame. These are going to be found generally in clusters along the channels, particularly at the large blood vessels, like we saw with the left subclavian artery, or excuse me, left subclavian vein of that one picture. These are going to be found particularly in the thoracic and abdominal cavities, where you have those larger blood vessels. You have some major aggregation points of these lymph nodes, particularly the axillary nodes, which are going to be under the arm. You're going to have the inguinal nodes, which are going to be where the hip meets the pelvis, and then the cervical nodes, which are going to be the ones at the neck. The spleen is much like a large lymph node, except instead of filtering lymph, it filters blood. And the spleen is what is going to filter pathogens from the blood. Now, you can live with your spleen, without your spleen. but your immune system is a bit compromised, and you do have to pay attention to some of the potential risks of a splenectomy. Sometimes you can't help it. You have to have the spleen removed, and the body system will go back to its normal working functional self. The thymus, this is going to be a gland where we have a number of things taking place, particularly the maturation of T cells. This is going to have a very high rate of activity throughout childhood and will slow down at about puberty. After you reach adulthood, it's going to begin to shrink, and as it shrinks, its activity also slows down. The thymus is going to release thymic hormones. This is going to help thymocytes that are going to develop and become mature T cells. In a baby, the thymus it sits right above the heart, and in a baby, that thymus is almost the size of the heart. And as we grow older, it shrinks down to about the size of an egg. And the parts of the immune response that the thymus is responsible for do tend to kind of slack off as we get older. You do have some other miscellaneous lymphoid tissue. These are also sources of the production of lymphocytes. Remember that any lymphatic tissue has the ability to produce lymphocytes. These are going to be your tonsils, breasts in pregnant and lactating women. You have these associated lymphoid tissues of the gut, the mucosa, the skin, and the bronchioles. You're going to find within the gut, these are going to include things like the appendix, the lacteals, and the Peyer's patches. All of the rest of these are going to have lymphatic tissues that protect different regions of the body, like the mucosa, the skin, and the bronchioles. Most of our immune response is going to originate in the blood. Everything but your first line of defense. The circulatory system is going to include veins, arteries, and capillaries. It also includes the lymphatic system. Within the circulatory system, we can divide our blood into whole blood or serum. Whole blood is going to be blood cells suspended in plasma that include clotting factors, whereas serum is going to be plasma without the clotting factors. Some of the characteristics of plasma are going to include that it is the main component of water. We have a lot of water in our blood, and when our body needs water, that's one of the places it's going to take it. And when you become dehydrated, the viscosity of your blood becomes thicker than normal. It does carry some proteins like albumin and some of these globulins like antibodies. It does have some immunochemicals that are very important in the processes of inflammation. It produces fibrin that's going to allow for some clotting factors that are going to help to isolate particular bacteria. Once we get into inflammation, you'll see about these clotting factors that are going to localize an infection. It does produce some hormones or secrete some hormones. It does contain nutrients as well as dissolved gases, and it is one of the places where waste products will go and be carried out of the body through the cardiovascular system. Blood is going to come from the cells of the bone marrow, and this is through a process called hematopoiesis. This is going to be the production of blood cells. Hematopoiesis is essentially the production of blood, of any type of blood cell. This is going to begin very early in embryonic development. You're going to begin to have blood cells beginning to be produced, and you end up then having cardiovascular circulation. This is going to be taken over completely by the red bone marrow when you have ossification of that embryonic bone tissue. If you remember your ossification lecture in A&P when you were in the bone section, and that when bone becomes ossified, it has the yellow marrow and it has the red marrow, and the red marrow is where hematopoiesis is going to take place. This is relatively short. What's going to happen is you are going to have then these precursors to new blood cells that are going to become that are stem cells in the bone marrow. This is when we're going to begin to take on some lifelong blood activities. These can become any kind of blood cell that they need to become. I do have a bit of an argument with your book, and I have emailed the author to get some clarification. In my learning about the immune system, the stem cells in the bone marrow were called hematopoietic stem cells, not pluripotent stem cells. What I want you to understand is the stem cells from the bone marrow can only become blood cells, not really any kind of cell that they want. The cells will differentiate ultimately in the red blood cells, the white blood cells, and the platelets, and these are going to be the mature cells. This picture right here comes from your book. You'll see this is the whole division and differentiation of blood cells. You're going to have stem cells that are going to divide. They're going to become particular stem cells, and then those stem cells will differentiate into their final competent cell. If we look here, oops, I'm sorry, I didn't mean to flip back on you. If you look at these hematopoietic stem cells here in the bone marrow, they will then become stem cells that are all going to be all of the blood cells except for the lymphocytes. And we'll talk about what the lymphocytes will come from here in a minute. So this stem cell can go any number of different directions based on something called chemotactic factor, conversion factors. These conversion factors are going to cause these stem cells to divide into a particular type of cell and then differentiate into that mature cell. So this stem cell can become a erythroblast, and that erythroblast will become an erythrocyte, which is your red blood cells. It can become a megakaryoblast. That megakaryoblast will differentiate, will mature into a megakaryocyte, and then as it gets older will fragment into platelets, and those platelets are going to be part of our blood clotting and inflammation. This stem cell can also become what is called a myeloblast. That myeloblast is going to be probably the most prolific of all of our immune cells. This is going to be the cells of our neutrophils, our basophils, and our eosinophils. These are what are called the granulocytes, and we'll talk about the difference between granulocytes and agranulocytes. Myeloblasts can also give rise to these things called mast cells, and these are going to be specialized cells that are going to trigger inflammation and allergic reactions. The last cell that these stem cells can become are the monoblasts. Monoblasts will become monocytes, and those monocytes will be triggered to become macrophages and dendritic cells. The monocytes will be monocytes when they're in the blood, and when they get fixed in tissues, they will be macrophages and dendritic cells. Going back up to the hematopoietic cell here, if it doesn't go the stem cell route, it's going to become a lymphoid stem cell, and that lymphoid stem cell will divide into our lymphoblasts, and those lymphoblasts will differentiate into our T cells and our B cells. The T cells and the B cells are going to be part of our third line of defense, and the lymphoid stem cell will also give rise to this thing called a natural killer cell. Now, I haven't talked much about the natural killer cell in this lecture, so I want to give you a little bit of information here. These are going to be cells that are cancer surveillance cells. These are going to be the cells that are going to take on any of the cells within the body that have refused to die in the process of their cellular cycle. This way, they can destroy those cells, and those cells will no longer become potential tumor cells. They're also very good at killing off any virally infected cells. Remember earlier I said that we could divide our white blood cells into the granulocytes and the agranulocytes. The granulocytes are large. They have very large cytoplasmic granules. These are going to be digestive enzymes, and they have a very lobed nucleus. These are going to be the cells that are the neutrophils, the basophils, and the eosinophils. Now, the way I remember this is I make the acronym BEN, B-E-N, is GRAND. So all of my granulocytes are going to follow into those basophils, eosinophils, neutrophils. The agranulocytes, these have very small granules within the cytoplasm. The nucleus is not lobed, but usually rather round. And these are going to be the T cells, the B cells, and the monocytes. The neutrophils, these are going to have a kind of a horseshoe-shaped nucleus. It's what they call a polymorphic nucleus. These are going to be the ones that are the most common in cellular circulation. You're going to have a high number of these in the blood as well as fixed in the tissues, and their job is phagocytosis. These are going to be very active engulfing organisms. They are going to kill mostly bacteria. Their job is to phagocytize bacteria. They're also going to degrade any of that phagocytized material. These are going to be the first cells to arrive at the area of injury, particularly in the inflammatory response. The next one are the eosinophils. These are going to be present in the bone marrow and the spleen. These are going to destroy eukaryotic organisms like fungi and worms. These are particularly invasive active in parasitic worm infections. These are going to be also associated with inflammation and allergies. These are the ones that are going to accumulate first at the site of inflammation. These granules are going to release specific enzymes called peroxidases and lysozymes. These are digestive enzymes that are going to go in and destroy whatever microbe they have phagocytized. Basophils, these are going to be in relatively short number. These are less than 0.5% of the white blood cells in circulation. These also have a lot of the same functions as the eosinophils, primarily in the inflammatory response and in allergies. These are going to release mediators like histamine, and these are going to basically when they become localized into tissues, they are going to become mast cells. These are going to be non-motile, and they're very important in allergic reactions and in responses of inflammation. The agranulocytes, these are going to be our B cells, our T cells, and our monocytes. These are going to be the lymphocytes are going to be second most common leukocytes that are produced outside of the neutrophils. These are going to work in your third line of defense. Your T cells are going to be cell-mediated immunity, and then your B cells are going to be antibody-mediated immunity. And again, we'll talk about this mostly in Chapter 15. The monocytes, these are going to be taken out from the bone marrow into the bloodstream, and these are going to live as phagocytic cells for a few days, and then they're going to differentiate into macrophages, which are going to be long-lived cells within the immune system. Their job is to be responsible for the nonspecific killing of any microbe that would get into the tissues. It's going to process foreign antigens. It's going to present them to the lymphocytes, your B cells and your T cells, and it's going to initiate your third line of defense. It's also going to secrete compounds that are going to either mediate, attract, or inhibit immune cells. So this kind of regulates the immune system. This is going to be kind of like the traffic cop of your immune system. You can go, you have to stay, you need to wait a little bit. That's the kind of message that this macrophage is sending. Then you have the dendritic cells. These are what they call relatives of the macrophages. These are going to be found throughout a number of tissues, particularly the tissues that have that reticuloendothelial system. These are going to process foreign matter, and they are going to present it also to the lymphocytes so that the lymphocytes can initiate an immune response and take care of the microorganism. These are pictures of where you would have different macrophages, particularly embedded into tissues. This top one here, this big round thing is a macrophage in the alveoli of the lung. Here is the alveolar cell, and this macrophage is actively consuming whatever this organism is right here. Here you have them in, these are what are called Kupffer cells. These Kupffer cells are the immune system of the liver. And then you have what are called Langerhans cells. These are the dendritic cells. You're going to find these in the skin. These are going to act as the immune mediators in the skin. So we've looked at our white blood cells. Let's take a minute to look at the red blood cells. Your erythrocytes are going to be your red blood cells, and they're going to develop in the bone marrow. They are going to lose their nucleus as they leave the bone marrow. Remember, when you looked at blood cells in A&P, you looked at the red blood cells, and they had no nucleus. And They have to have a nucleus to divide and differentiate in the bone marrow, but once they are maturing, they will lose that nucleus and they will enter into the systemic circulation. Their job is to transport oxygen and carbon dioxide from the tissues, and that's essentially their whole job. We talked about the megakaryocytes. Those megakaryocytes are going to fragment and become platelets. So these are actually formed elements in circulation, not whole cells. They're just going to be bits and pieces of a cell. These are going to function in the homeostasis of the circulatory system. They're also going to release chemicals for blood clotting and in the inflammatory response, and they are going to be responsible for the blood clotting within both inflammation and circulatory homeostasis. Remember I said we were going to talk about specifically the four mechanisms: inflammation, phagocytosis, interferon, and complement. Well, here we are. We are going to talk specifically about each one of these individually, and all of these are going to play a major role in the second line of defense. The inflammatory response is going to be any reaction to any kind of traumatic event to the tissues, whether it's a tiny injury like a thorn prick you see in this picture, or some sort of massive injury that is going to initiate an inflammatory response. You have the classical signs and symptoms. You've got what is called rubor, which is redness. You've got calor, which is heat. Tumor, which is swelling, and then dolor, which is pain. There is a fifth symptom that is essentially the loss of function. So if you think about it, you've got heat, pain, redness, swelling, and loss of function. Those are the 5 cardinal signs of inflammation. In the inflammatory response, you're going to have the mobilization of a number of components of the immune system to the area of injury. Once you set these mechanisms in motion, you're going to begin to repair tissues that have been damaged. You're going to localize and clear away any foreign debris, any foreign microbe that might be there, and then you're going to destroy any microbe that has invaded, and you're going to block their further movement into cellular tissues. If you look here, this is a picture from your book. Here we have our injury. That injury is going to come through, puncture the skin, and in this case it actually punctures the capillary. This is going to cause a number of different things to happen. First, you're going to have the mast cells release these things called chemical mediators, and that's going to initiate the vasoconstriction of the blood cell. You're going to have a nervous response, and that's going to cause a minor or a rapid vasoconstriction where you're going to kind of cut off any blood flow there. This is going to trigger some vascular reactions then. You're going to start to form a clot to try and eliminate excessive blood loss. Then any bacteria in there are going to initiate the production of these neutrophils. You're going to have seepage of plasma and white blood cells out of the blood vessels, and those vessels then, after they've had that momentary vasoconstriction, are going to have vasodilation. As they dilate, these areas in the blood vessel are going to separate, and they are going to open up for these white blood cells to seep into the tissue so they can go and do their job at phagocytosis. After you have some of these vascular reactions take place, one of the things that's going to happen is you're going to have the production of edema, the swelling. You're going to have a bit of pus formation, and you're going to have a significant number of neutrophils, and then this fibrous exudate. We're going to talk about what this exudate is, and you're going to end up then with this pus formation. Once all of this takes place, you're also going to begin to scab over the area where the clot initially formed. Once you have this third stage has kind of run its course, it's going to move into the resolution and healing stage of scar formation. You're going to have lymphocytes that are going to come out of the blood cells. You're going to have macrophages coming out of the capillaries. You're going to have scar formation over the surface of the injured tissue. This is going to be new tissue that's going to take place within the first 24, 48 hours of the injury. You do have some early inflammatory events. If we go back, if you flip back in your book to the picture that we just looked at, you're going to have this controlled nervous response that's going to release those chemical mediators. Remember, those mast cells are going to release those mediators. They're going to release something called cytokines, which are going to be taken out by the blood vessels in the tissues. You're going to have tissue cells that are going to be activated, and then platelets are going to come to that area of injury and try and wall off that area so that you can keep those microbes localized. You're going to have some mediators that are going to affect the endothelial cells of the smooth muscle of the blood vessels, which is going to cause that constriction, and then we're going to start with our vasodilation. You have some chemotactic factors, or called chemokines. These are going to affect white blood cells. These are going to be things that are going to draw those white blood cells to the area of injury. You're going to have the production of fever. You're going to have some of those lymphocytes are going to be stimulated to come to that area of injury. Hopefully these are going to prevent viral spread, and you're going to have some stimulation and potential cause of allergic symptoms. The arterioles next to that area of injury are going to be constricted very quickly and then vasodilate. And when they vasodilate, you're going to start having that increase of white blood cells moving out of the tissues moving out of the blood cells into the tissues, where they can go in and clean up the damage. Right in the middle of the whole inflammatory response, when everything is taking place, this is when you're going to have that leakage of that vascular fluid into the tissues. This is essentially called edema. Essentially what happens is you have exudate, things that are leaking or escaping from the gaps in the veins and the venules. They are going to accumulate and cause edema. This is going to contain this edematous fluid is going to contain plasma and plasma proteins, as well as blood cells, and even some of the cellular debris that was created by the damage. You can have this edema be clear, where you have very little microbial activity in here. This would be what we call the serous fluid, or it can be contaminated with red blood cells or pus, and that is going to be all of that white blood cell activity, where you have cells that have kind of died, white blood cells, regular tissue cells that make up this pus material. You have two mechanisms that are going to take place during this kind of intense inflammatory stage, and that's called chemotaxis and diapedesis. Chemotaxis is where the white blood cells are going to migrate into the tissues in response to some sort of chemical stimulus. Those mast cells releasing those chemical factors into the tissues. That's going to call those white blood cells to migrate out of the veins into the tissues. Once you have those chemotactic factors released and those white blood cells start to leave, when they leave the vessels through those separations in the tissue spaces, then you have something called diapedesis. And once those white blood cells then get into the tissues, then you can go in and you'll have a very rapid immune response that's going to try and control everything that's taking place. So if you look here, here's a great picture of diapedesis and chemotaxis. You have those chemotactic factors that are released by the mast cells. Those are you've had your vasodilation as a part of the inflammatory response, and now you've got these blood cells that are going to start squeezing through these openings that come from the endothelial cells of the blood vessel. When those cells migrate out into the tissue space, then they're going to go in and start dealing with the microbes that are potentially causing harm. If you look here, this is actually a micrograph. Here is the blood vessel. Right here, you've got all of these cells that are coming through here, and this is basically an opening in the endothelial lining, and this cell is squeezing out into the extracellular space so it can go and deal with whatever microbe has invaded the area. You do have some benefits to that edema. That fluid can dilute toxic substances that get released by microorganisms. You can also have those platelets coming in, and they are going to form those fibrin clots and trap those microbes and localize them to that tissue. Phagocytosis is going to occur immediately as those white blood cells leave the blood vessels into the tissues, and you're also going to have certain bacteria that are going to produce what are called pyogenic or pus-forming chemicals that are going to attract neutrophils to the area, and they're going to go in and consume any pathogen that is in the way. We talked about the early reactions, and then you have those edema reactions, which are right in the middle, and now we have the late reactions of inflammation. This is a more long-lived inflammation that is going to attract a collection of cells like monocytes, lymphocytes, and macrophages to the area that you have the injury. Macrophages are kind of like the guy at the circus that goes behind the elephant with the broom and the trash can. Macrophages are going to clear away any of that pus, any cellular debris, damaged tissue, dead white blood cells. It's going to kind of clean up the area. Then you're going to have B lymphocytes that come to an area that are going to produce antibodies. These antibodies then are going to initiate other cellular responses that are going to allow for some other processes to take place, like complement, that are going to kill the cell. And then you're going to have T lymphocytes that are going to go and they're going to kill any intruder directly and destroy what's left in the tissues. These late processes are going to initiate repair or replacement of connective tissue, and this is where that scar formation is going to take place. In the inflammatory response is pretty remarkable in that you almost have repair almost as rapidly as you do injury. Depending on the injury, if the injury is mild, then this inflammatory response doesn't take very long. If you have more damage, then you may have an inflammatory response that may go days or even weeks, but eventually repair does take place. Sorry, I didn't mean to skip that one. This should be an animation. I'm hoping that it comes up. It's not going to. I will place the animations that don't come up in your either your My Courses, or I will send them to you via email so that you can click on them and watch them. The next mechanism that I want to talk about is phagocytosis. Phagocytosis, in my opinion, is a much easier process to understand than inflammation because it is a very step-by-step process. Basically, phagocytic cells are going to survey any type of tissues. They're going to look for microbes. They're going to look for anything that is not normal host tissues. It's going to also look for injured and dead cells, and it's going to go ingest them and eliminate them so that they don't cause damage. You're going to have some what they call immunogenic information that can be taken in by these macrophages, or excuse me, through the process of phagocytosis, and then they can present that immunological information on their surface and initiate another immune response. These things that they are presenting or extracting are what we call antigens. There are three main types of cells that are going to be your phagocytic cells. These are going to be your neutrophils, your monocytes, and your macrophages. Your monocytes are going to be able to phagocytize, and they have essentially three different stages in which they're going to have their differentiation and maturation. Within the bone marrow, you're going to see that you've got your stem cell here that is going to become a promonocyte. Once it leaves the bone marrow and gets into the circulatory blood flow, then it becomes a monocyte. Once it leaves that circulatory blood flow and gets into the tissues, then it is called a macrophage. Their jobs are the same. They are going to be phagocytic cells, and you're going to see the macrophage is going to play a very significant role in the immune response of your third line of defense. The stages of phagocytosis are going to include this stage of chemotaxis. Chemotaxis is going to be where you're going to have particular antigens releasing what are called pathogen-associated molecular patterns. And these are going to be signals that are going to be sent to the white blood cells, those phagocytic cells, and they are going to come and they're going to bind to specific receptors on the surface. And these receptors are called toll-like receptors. And these toll-like receptors are going to bind to these associated molecular patterns, and they're going to then attach the microbe to the white blood cell. Sorry for that interruption. Once you have the microbe binding to the receptor on the neutrophil, that, or on the phagocytic cell, whether it's a macrophage or a neutrophil or a monocyte, whatever, this is going to engulf those cells and they're going to ingest them. When they ingest them, they're going to form this vacuole inside the cell called a phagosome. That phagosome is essentially a compartment that holds that microbe in it. Okay? Once that phagosome gets into the intracellular space, it binds with something called a lysosome. That lysosome has those degradative enzymes in it. They bind to the membrane of the phagosome, and it produces something called a phagolysosome. The lysosomes are going to dump their digestive enzymes into that phagolysosome. They're going to destroy the bacteria, and once they do that, then that phagosome is going to bind to the surface of the membrane of the cell, and they're going to eliminate the waste that is the result of the destruction of the microbe into the extracellular space, where it would be picked up and taken out through some sort of waste elimination process. If you look at this picture is on page 415 in your textbook. You have your, here you have your particular phagocytic cell, whether it's a neutrophil or a macrophage or whatever, and you have these bacteria out here. These bacteria produce those pathogen-associated proteins that are going to be all on the surface of those bacterial cells. That chemotaxis is going to cause adhesion of these bacteria to receptors on the membrane of the particular phagocytic cell, whatever that might be. That's going to cause this cell then to have this invagination produced, and once that invagination happens, these pseudopod type arms here are going to come and fold up against each other, and they're going to produce this phagosome. Okay, this phagosome is going to then be what is enclosing all of these microbes. These microbes are going to form or going to fuse, excuse me, with these lysosomes. These lysosomes contain these degradative enzymes here, and they're going to fuse with that and release their digestive enzymes into the phagosome. Once they have fused together, now we have what is called a phagolysosome. Okay, that is going to degrade all of the cellular contents here, and now we have killing and destruction of the bacterial cells within this phagosome. What we have left is essentially residual debris that will fuse with the membrane here and be released through a process of exocytosis. Here are a number of different enzymes within the lysozymes. You don't have to know these, just understand that these are going to be digestive enzymes that are going to be released in the process of cellular degradation. I think this is an awesome picture here. This is some sort of macrophage or phagocytic cell. Here you have all your bacilli here that are being consumed by this phagocytic cell. I said something about surface receptors. You've got your those processes where you have your bacterial cells binding to pattern recognition receptors on the host cell, and I said something about these toll-like receptors. Toll-like receptors are going to be surface receptors that are going to bind to particular molecular patterns. They're going to send out a signal. They're going to send out these cytokines, these interleukins, inflammatory mediators, and this is going to initiate any number of things. It can initiate things like phagocytosis, or it can produce inflammation. All of these things are going to lead to some sort of destruction of the microbe. This toll-like receptor here is essentially going to cause when it releases these cytokines, and this is going to induce then phagocytosis, and this cell will go in through the phagocytosis process and destroy the microbe. Apparently this animation is not going to come up either, so again I will send these to you via some other method. Let's see if phagocytosis will play for us. Nope, it's not going to play either. So again, I'll get them to you, and the one on the cell receptors, I don't believe I will be sending to you. It's kind of a convoluted cell receptor, and it doesn't really help you out much. So just look for some of like the phagocytosis, inflammation. Those two are relatively good. Fever is one of the things that can be part of the inflammatory response, or it can act as an adjunct to the inflammatory response. A fever is any elevated temperature that is abnormal to the body. Essentially, when we have these inflammatory fevers, these are what we call fevers of unknown origin. To initiate a fever, you're going to have some sort of pyrogen that is going to set some sort of mechanism in the hypothalamus to change the thermostat to a higher setting. When that thermostat gets set to a higher setting, then you're going to have the body temperature rise. Muscles are going to increase their heat production. You're going to have the arterioles in the peripheral blood flow, the arms, the legs, places like that, is going to decrease, and therefore heat loss is going to be minimized because of vasoconstriction, and then all of that heat is going to go towards the core body temperature, and the core body temperature then is going to increase. Any pyrogen can be exogenous or endogenous, meaning it can come from the outside or from the inside. I just want you to understand that fever is sometimes a very good thing. This can be something that works with the inflammatory response and help with the whole inflammatory process. You do have some benefits of fever. When you've got microbes that are temperature sensitive, these fevers that go clear up to 100, 101, 102, these microbes are going to perish in body temperatures that are that high. It can impede the nutrition of the bacteria by reducing the availability of iron. At a fever, you're gonna hold on to that iron at the hemoglobin. The hemoglobin is going to hang on to it, and bacteria are going to have very little access to it. It can also increase metabolism and stimulate the immune response, and it's going to cause some naturally protective physiological process that is going to essentially be a good marker to help with the inflammatory response. So fever, if it doesn't go beyond, say, 101, 102, is a good thing. When it goes beyond, when it gets to about 102, 103, then you need to do something to bring that fever back down. Fever is one of those homeostasis mechanisms that is a self-amplifying system. This is a positive feedback mechanism. So again, you have to do something to break that continued increase in fever. But if the fever is 99, 100, 101, these are relatively good fevers, and you should just deal with the symptoms and allow the fever to take its course. Interferon, these are antimicrobial proteins that are produced by the body, particularly against viral infections. It's also a regulation of the immune response, as well as some intercommunication between other tissues. We have three types of interferon: interferon alpha, beta, and gamma. All three of them are going to be produced in response to viruses, in response to RNA that is abnormal in response to particular immune products that are initiated during some immune response like phagocytosis or inflammation. And these are also going to be produced in response to a number of different antigens, particularly viruses and bacteria. These interferons are going to bind to cell surfaces. They're going to induce some sort of genetic change in the expression, and the bacteria or the virus is going to die. This can also inhibit the expression of cancer genes. It can turn those genes off, it can change the genetic expression, and it can turn off any kind of potential tumor production, and you have then tumor suppression that can cause these cells to die off and not produce the genes that they are coding for. So if we look at this picture is in your book on page 418. You've got this virally infected cell here, and that virally infected cell is going to release an interferon. It doesn't really matter which interferon, but this interferon is going to attach to specific receptors on a nearby uninfected cell. The infected cell is going to die anyway, so it's going to release this interferon kind of as a last dying gasp to save the neighboring cells in that same tissue. So when the neighboring cell gets infected by this virus, depending on the interferon, that interferon can activate particular genes, and then those genes are going to synthesize antiviral proteins that are going to destroy nucleic acids. They're going to destroy messenger RNA. They're going to destroy any type of viral replication, and this cell then is going to be saved at the expense of the infected cell. So interferon activity can essentially save neighboring tissue while in the process of the original cell that's released the interferon has died. Let's try this one too. Nope. Antimicrobial complement. This is going to be a series of cascade proteins that are going to work together to destroy bacteria and certain viruses. This is kind of a domino reaction. This reaction is going to have 3 different pathways, and each one of these pathways is going to result in the puncturing of the cell membrane. You have the classical pathway, you have the lectin pathway, and then you have the alternative pathway. The classical pathway is what I am going to talk to you about. Just understand that the lectin and the alternative pathways end up causing the same damage, but in a different beginning. If we look here at the classical pathway, its activator is going to be some sort of antibody that binds to the cell and initiates it at the very beginning at what we call C1 complex. Lectin, this is going to be a pathway that starts a little later down the line. It's going to be activated by molecules called mannins. These mannins are going to bind to mannose binding lectin, which is going to then initiate the complement proteins, which will end up going to the final process of putting the holes in the membrane. And then you have the alternative pathway. This is going to be bacterial or fungal cells, viruses, as well as parasites. These are going to be initiated at the binding site on the part of the complement protein C3. That C3 is going to combine with some other factors that are going to lead you to the same membrane attack. These membrane attack proteins C5, C6, C7, C8, and C9 are going to insert proteins into the cell that are going to cause a hole to be formed or a pore to be formed in the membrane. And so these are called the MAC attack complex proteins, or the membrane attack complex proteins. If we look here, the first step that we have in the complement cascade is through this is the classical pathway. This is going to be initiation, and this is where you're going to have C1, that C1 complex protein binding to your antibodies. The second step is going to be amplification, and cascade. This is kind of all happen together. When that C1 complex is activated, it's going to activate complement protein C4 and C2. So we have, let me find my arrow here, C4 and C2. These are going to be cleaved and separated into C4A and C4B and C2A and C2B. The A fragments, C2A and C4A, are going to act as inflammatory mediators. Okay? Once we have the cleavage of the C4 and C2 proteins, they're going to bind to C3. C3 is going to be enzymatically cleaved into A and B fragments, and it's also going to act as an inflammatory mediator. Once we have C4B, C2B, and C3B together, this is going to form an enzyme that's going to activate C5. This is going to be the end of that amplification cascade mechanism. Once we have that C5 cleaved into, let me find my arrow again, C5A and C5B. C5A goes by the way of inflammation, and C5B is going to be the protein that has the reactive site for that assembly of that attack, membrane attack pore formation. C5b is going to initiate the activation of C6. C6 is going to activate C7, and C7 will activate C8. All of this from the activation of C5. Once you have the formation of that, then you're going to have the activation of C9, and this can cause the formation then of these pores. So C5, C6, C7, and then C8 is going to activate the insertion of these proteins within the membrane, and these are the membrane attack proteins. There can be up to 15 of these to form this particular membrane here, this membrane attack complex. Once you have that polymerization of that membrane attack complex, that membrane attack proteins, then you have the full membrane attack ring, and this is going to give you kind of that donut shape that's going to puncture very small holes in the membrane. And once you have all of these little holes within the membrane, all of these little black dots are places where you have, this is a big enzyme that is essentially going to cause these to form holes, and everything now on the inside is going to flow out, and anything on the outside is going to flow in, and this cell is eventually going to lyse and be destroyed. Another animation that'll be coming your way. One of the other things we need to look at a little bit is the ability of bacteria to bind to iron. Iron is a very important component to microbes and to humans, and we have a number of different iron binding molecules that are going to lessen the ability of these bacteria to grab hold of that iron, so it's not going to be available. This is probably the most important. Hemoglobin, transferrin, and lactoferrin. Hemoglobin is an iron binding protein that is going to keep that iron away from bacteria. It's going to bind tightly, and this hemoglobin is going to be located in the red blood cells. We all are very familiar with red blood cells. Then you have transferrin, which is going to be found also in the blood and tissues, and then lactoferrin, which is going to be found in breast milk and saliva. All of these are going to do the very same thing. They're going to bind iron, they're going to sequester it, and they're going to make sure that these bacteria are going to have to fight very, very hard to get this iron, and it sequesters it for use for the body, and it leaves it away from the bacteria. Some bacteria produce these things called cytophores, and these are basically going to grab away any type of bound iron. These are capable of scavenging, however you would say that, iron from any kind of iron binding protein like the hemoglobin, transferrin, and lactoferrin. You can get these, and it kind of acts like a tug of war between these binding proteins and the bacteria, and eventually you are going to get that those bacteria may eventually get that iron off. If you remember from when we talked about fever, these temperatures make iron even less available to pathogens. These proteins kind of hang on to them even tighter so that these bacteria can't get them. So you can see that, remember I said that the immune system was complex. It's not really complicated, but it's very complex. You've got a number of different ways. Microbes, it's kind of amazing that microbes get a handle on us at any given time. You can see that if our immune system is down, we're going to have some problems maintaining our ability to stay well and to keep pathogens from getting a stronghold. But if we eat right, if we sleep, if we keep our immune system healthy, you can see that it's going to be very difficult for a pathogen to jump in there and try and make us sick. I will be producing chapter 15 here in the next day or so. It will be available for you online, and please be sure that you either in class or through an email get any questions that you have over this material to me as quickly as you can so I can get information back to you. Thanks a lot, and I'll talk to you later.